Alright, let's get right into this. We got no time to waste. First of all, let's learn. This is Unit 1, Chemistry of Life. So, we got a null hypothesis. These are just a few terms you'll need to know. So first of all, what's a null hypothesis? Well, in a problem where there's situations, a null hypothesis is when the variables, there's no correlation between them in that certain situation. And next is an alternative hypothesis. It's basically a null hypothesis, but there is a correlation between the variables. So if one thing is affected, it affects the other. Now, an independent variable, a factor in a problem that doesn't change. No other factors change it. So basically, it stands alone. Nothing changes it. It's on its own. A dependent variable is basically when a factor that is affected by another factor. So a variable affected by another variable, it depends on the others, hence the name. A control group, that's just the original test of the experiment. A control group, a test that's not influenced by other factors. So basically, it's what you're originally using to set a basis for everything else. So let's talk about water and its characteristics. First of all, the recipe for water is pretty simple. It's 2 hydrogen atoms and 1 oxygen atom. Pretty simple. And the atoms are held by polar covalent bonds. Now, what are those? Well, a polar just means electrons or charges around the molecule that ain't equal. So if you notice, there are 2 hydrogen atoms and 1 oxygen atom. That's 2 to 1. And to add on to that, the electrons, the charges surrounding the molecule, are oxygen, which is a negative charge, and hydrogen, which is a positive charge. So there's 2 positives, 1 negative, which makes it unequal. Alright, now from the last slide, we gotta first know, what is polar and non-polar? First of all, polar is the uneven spread of electrons, or the charges, around the molecule. Non-polar is an even spread, the exact opposite, on the molecule. And just like it says, opposite forces attract. So if you notice in the picture, the hydrogen, which is negative charge, and the oxygen, which is a positive charge, attract to each other. Just like a magnet. So that's a good way to remember. Now let's learn about cohesion and adhesion. So first of all, a cohesion is basically when molecules are connecting with the same type of molecule. So like water. Water wants to connect with itself. Now adhesion, molecules connecting with anything else. So if you see in the picture, the mercury forms beads on the surface because of the cohesion, because they're different substances, they don't match together well, but it will still attract. Now let's learn about some organic and inorganic compounds. Organic compounds are carbon and hydrogen bonds. So those are like the general basis. Now inorganics are just ionic bonds. So what's an ionic bond? It's a bond between outer electrons and other molecules switching electrons with each other. So as you can see that the Na plus is switching to the Ci minus. Now bonds are just 2 or more atoms / molecules or just molecular bodies combining with each other. Now, here's a big part of this unit, macromolecules. Now what are those? They're just the building blocks for life. So there's 4 types, carbohydrates, lipids, proteins, and nucleic acids. And they're characterized by these elements on the periodic table, like CHO, carbon, hydrogen, oxygen. Same thing for lipids. And then there's nitrogen for proteins and nitrogen and phosphorus for nucleic acids. These are basically what they're constructed of, and they play in bigger roles later on. Okay, so let's get into lipids. So they're basically made from fatty acids. And now what are fatty acids? Well, they're small units of fat, wax, and oils. Now there's 2 types of fatty acids, saturated and unsaturated. So saturated have chains of hydrocarbon bonds, which is just one continuous bond of hydrogen and carbon atoms, as you see, the H and C. Now unsaturated, they can have a double bond or more of them in the middle, which separates them into this little elbow shape. Alright, let's talk about proteins. So proteins are made of amino acids. Now, whoa there, what are amino acids? Well, they're just the building blocks for protein. Wow! So there's 4 structure levels of them. The primary structure, a chain just of amino acids. It's like a bead bracelet kids make it, and what you make as kids. Secondary structures are when the chains of amino acids are coiled or folded up together, as you see in the second part. Now, tertiary. the shape of the whole amino acid tangles as groups. So that whole, like, those folds and stuff just tangle up into one big structure. Quaternary are basically multiple of those tertiary structures combined together. Okay, so let's talk about nucleotides. Now, what are they? Well, they're also known as nucleic acids. Well, they're basically 4 bases of nitrogenous bases: adenine, guanine, cytosine, and thymine, a phosphorus, and a sugar. And to create all of this together makes deoxyribose. So a DNA is double helix with thymine. RNA is a single helix with uracil. And these pentose sugars create those DNA structures. Okay, now we're in Unit 2, Cell Structure and Function. Now, let's talk about ribosomes. So the recipe to create ribosomes are RNA and protein. So ribosomes go through the RNA and create the protein, in which you can see in the image, mRNA goes through the ribosome and creates the amino acids. Now, ribosomes read the mRNA, the messenger RNA, and create the amino acids, the production of the 4 structures we saw earlier. Okay, so let's talk about the endomembrane system. So this system creates, changes, and carries out proteins. And it's made of these organelles, as the Golgi apparatus, rough ER, smooth ER, nucleus, and the vesicles. So the rough ER creates and ships the proteins to the Golgi. The smooth ER cleans the cell, makes sure it's not dirty. The Golgi is basically like the postal service. It transports out the proteins. The vesicles are basically lysosomes. They're like the carry trucks for those packages that the Golgi sent. And the vesicle carry trucks transport the cell waste out of the cell. Now we're at a part I don't like. Surface area. So let's get this out of the way. Smaller cells can diffuse better. If you don't know what diffuse is, go search it up. So the steps to know the surface area: first of all, know the surface area, which we got a nice diagram here. Then you got to know the volume. Then you create a ratio between them. So just look at the problem on the right and you'll get a general idea on how this works. Okay, so let's talk about the plasma membrane. Back to science. The plasma membrane, to make it, are phospholipids. And if you notice, the word phospholipid has phosphorus and lipid in it. Now, to make phospholipids, you need a glycerol and a fatty acid. It has a hydrophilic head. Now, that lets water into the wall. Hydrophobic tail doesn't let the water into the wall. Now, a protein channel allows certain molecules to enter and leave, but not others. Now, getting in and out of the cell membrane is a little bit more complicated, so let me explain that. So, diffusion from the previous slide. Diffusion is basically a molecule moving from one area, a concentrated area, to another. So basically, just a higher concentration moving to lower concentration, or vice versa. Passive transport is a molecule going through the membrane without any energy needed, basically higher to lower. Active transport are molecules going through the membrane that need energy, which is lower to higher. Facilitated diffusion is spontaneous passive transport, so just all at once. Simple diffusion is a molecule going through the semi-permeable membrane, so that means like it lets some things in but not others. Aquaporins are just channels to help proteins come and go. Now, endo and exocytosis. Endo is capturing the substance with the membrane. Vesicle is going through the membrane and combining with the phospholipids. Okay, finally, something easy. So, osmolarity is the concentration of solute, and water potential is the potential of water on how pure the water is. So, that thing on screen. It's basically when membranes separate organelles. So basically, what keeps the organelles separate from each other? It's the walls, and that's what enzymes do. So now, endosymbiosis, that's a really cool thing. It's a theory that explains the evolution of eukaryotic cells from a single-cell organism. Okay, so now we made it to Unit 3, Cellular Energetics. Okay, so let's learn about enzymes. So enzymes are just a type of protein. A substrate is an activator for a chemical reaction of an enzyme. A product is the end result of that chemical reaction between the substrate and enzyme. So, on an enzyme, there's something called an allosteric site. It's where the inhibitor bonds and changes the enzyme. And now we'll learn about a competitive inhibitor, taking the substrate's place in the active site. So it basically just takes the place where it's supposed to be for the substrate. A non-competitive inhibitor is the inhibitor binding to the allosteric site and changing the enzyme to where the substrate can't bind to it. Now, exergonic and endergonic. So when exergonic is when the chemical reaction requires energy to kick off, or the exergonic just means releasing the energy. Okay, so I didn't really explain what denaturing was, but here it is. So denaturing the enzyme, changing or removing the qualities of the enzyme. So denaturing factors of it could be if something's too hot or too cold, the pH is too acidic or alkaline. You know how we drink water and they say the pH is too much or too little. So here are some reaction rate changes. The temperature increasing or decreasing it, the pH changing how acidic or alkaline the enzyme is, or the substrate concentrations. How much of the substrate is there? And you notice that the denaturization makes the substrate unable to bind to the enzyme. Okay, so something simple is photosynthesis. So it's the two processes within the chloroplasts. So first of all, light reactions. As you can see on the left side of the chloroplast, creates ATP and NADPH. It uses light and water to cause the reaction, and a byproduct is oxygen, which we breathe in. Now the Calvin cycle, it creates NADP plus ADP. And it uses the carbon dioxide we breathe out or maybe emissions. Also, it makes glucose as a byproduct, which helps the plant survive. Alright, okay, this is going to be more difficult, so let me take this through. So cellular respiration. The first process of cellular respiration, first of all, happens in mitochondria, is glycolysis. It converts glucose into 2-pyruvate. And there's two processes of it, aerobic and anaerobic respiration. Aerobic is just creating energy with oxygen. Anaerobic is creating energy without oxygen. That's a type of it, you'll see. Anaerobic respiration leads to fermentation, and fermentation is converting sugars to acids. Now let's skip on to aerobic respiration. After glucose is converted, it gets turned into acetyl-CoA enzyme A. This leads to the Krebs cycle, which I'll explain. The Krebs cycle produces 2 ATP energy. Then the Krebs cycle leads to electron transport and chemiosmosis. It creates the ETC, it goes through the ETC, which is a chain of proteins that guides electrons through the cell membrane. Okay, so let's finish up the last slide. So chemiosmosis is when ions move down the concentration gradients from high to low, low to high, same thing. So, and lastly, the oxygen is used to expel carbon dioxide from that previous reaction. Now, the electron transfer chain and oxidative phosphorylation. So ETC, how it works is when the electrons pass through the channel, it releases through redox reactions, which releases energy. Now, what's a redox reaction, you say? So it's a chemical reaction when the electrons pass through the ETC. So it goes through and produces energy. Now, what's oxidative phosphorylation? It's basically when electrons that came from the NADH and the FADH2 combine with the oxygen, and this speeds up the creation of synthesis of ATP from ADP. Okay, everyone, now we're moving on to Unit 4, Cell Communication and Cell Cycle. We're halfway there. Okay, so cells communicate through signaling. So here are the types of signaling. There's autocrine, which secretes hormones based on the receptors of the cell. Juxtacrine, contact between cells between binding. As you can see in the picture, they're binded together and they're communicating. Paracrine are cells when they communicate through a short distance. So they're near each other and they send their information to each other. Endocrine is when hormones sent through the circulatory system. And you'll see this happen, like you'll know a lot about this in other classes too. Okay, let's keep the ball rolling. First of all, we got types of junctions. So tight junctions are when they seal to other junctions tightly. Anchoring is when they connect two cells together but not fuse them together. A gap junction connects the cells through a protein channel, which we learned about before, allowing communication between the cells. Now, let's go to the next slide. Stages of cell signaling. Reception, when the cell detects a signaling molecule from the outside cell. So basically, when something activates it, it'll send a message to the cell. Transduction, it's making the signal more prominent, like it can amplify it, make it bigger. Now, response is when the signal is carried out, when it's followed through. Okay, so let's see how the cell responds through cellular response. First of all, it's how the cell responds when the protein connects with the receptor. So, in signal transduction, protein kinase activates once a phosphate group is added through the phosphorylation cascade, which is that like a staircase looking thing. So the first messengers are the ligands, the secondary messengers are the C and calcium ions along with inositol triphosphate. Okay, so let's learn about what G proteins and the changes in the signal transductions. First of all, G protein is just guanine nucleotide binding proteins. So they're guanine nucleotides that bind. So in those change include how proteins can activate based on receptors, and sometimes those changes can be bad, which sometimes they have instructions for something else, like a virus. Okay, so here's something simple to learn. Feedback loops. So a positive feedback loop is when the body activates an increased stimulus it feels. So like if you get a cut, your body will send platelets, an extra rush of blood, to that area instead of, slowing it down. So negative feedback loop is when the body tries to negate the effect of that stimulus. So like if you're too cold, it will try to warm you back up. And this is all ties into homeostasis, which is the balance in the body's vital signs. Okay, so let's get to mitosis and cytokinesis. So mitosis is the process where non-sex cells split. Now, I have a video explaining this better, so go watch that. Now, cytokinesis is when there's too much of cytoplasm and you get used to bring the separation of the daughter cells. As you can see, that's where its separation happens in the picture. Okay, so let's learn about cell cycle regulation. So G1 phase is where the cell is growing, getting those materials. S phase is when the genomes get replicated. G2 phase is preparing to divide. Mitosis, which is, I explained that. And cytokinesis is when the two cells split. Now, from the last step, there are checkpoints to these phases. So G1 checkpoint decides if the cell should divide. The G2 checkpoint triggers the start of mitosis, and the M checkpoint ends the dividing of mitosis, so the splitting of the cells. Let's go! We're in Unit 5, which is heredity. We're almost there, 3 more units. Okay, so let's learn about meiosis. Well, meiosis only happens in gamete sex cells. So it happens during interphase where diploid cells form, which those have 2 copies of chromosomes. Meiosis 1 is where chromosomes and cells split. Meiosis 2 is when the chromatids from each other split into 4 haploid cells, so 4 of those chromatids. Alright, so let's find the sources of genetic variations. Genetic recombination. Sister chromatids randomly distribute. So like no 2 siblings are ever the same. Random fertilization is the random chance of what genes just are. Random mutation. Now that's interesting. It's the random changes in genes causing the mutation. So you don't know what it could be. It can be either good or bad, or just have a trait in the family. Alright, let's learn about Mendelian traits. So, a gene is basically a functional gene of heredity and inheritance, passed from a parent to an offspring. An allele is a trait characterized by the ends of the chromosomes, a specific version of a gene. A locus is the location of a gene on the chromosome. A phenotype is used to determine physical characteristics of an organism, like the eye color, hair color, skin color. Genotype, the genetic makeup of the organism, just what they're made of, generally. Okay, so let's learn about more Mendelian traits. So first of all, dominance. They're traits that are overshadowed that overshadow recessive genes. Segregation. When 2 gene copies split randomly to create a new factor. So like, when the 2 gene copies split, there's a new factor that's included. Independent assortment. Pairs of traits that split during meiosis independently. Okay, so next is non-Mendelian traits. So one of them are incomplete dominance. It's a blend of the parents' traits in the heterozygous offspring. So as you can see, red and white make pink. So those are the 2 traits combined. Now codominance is when both traits are presented in the phenotype. So as you can see on the left side is the white and red both being presented. So red and white traits are both expressed. Now, sex-linked traits are genes found on the X chromosome. This affects males and females differently based on just gene characteristics. Okay, so recombination and linked genes. Recombination is the new combination of genes, so that's the randomization of traits. Now, what are linked genes? The closer the genes are, the more likely they are to be inherited. So that's how the males and females are during the sex-linked traits. Now, how do you find distance of gene mapping? Use the percentage of the offspring between the generations, and then you should find your answer. Okay, so let's talk about non-nuclear inheritance. So just means inheriting genes not from the nucleus, but from another origin, such as cytoplasm or the mitochondria. That's what mitochondrial inheritance is. They can also be shown in pedigrees as different patterns. Now, gametes and non-disjunction. Gametes are just sex cells, and non-disjunction is the failure of the oh my god, did I read it as that? Homologous chromosomes, or sister chromatids, to separate during meiosis. Now we're on Unit 6, which is gene expression and regulation. Okay, to start off with this unit is the pyrimines and the pyrimidines. The purines are adenine and guanine, and pyrimidines are cytosine, uracil, and thymine. They are nitrogenous bases, and purines always pair with the pyrimidines. Purines are bigger structures than the pyrimidines. Okay, so now DNA and replication. This is a bigger unit, bigger section, so just have to bear with me. So DNA replication is just making more DNA. It happens in the nucleus, not in prokaryotes. That happens in the cytoplasm. It has helicase, unzipping enzyme, the DNA polymerase, which is the DNA builder, primase, the activator, and ligase, the glue. Helicase first unzips the DNA from 5' to 3'. Topoisomerase stops DNA from recoiling. Primase places primers made of RNA. DNA polymerase starts building the new strands from 5' to 3'. Ligase fills in the Okazaki fragments and glues DNA together, and these two DNA strands are semi-conservative. Okay, so DNA replication is more complicated, so let me explain a few details. 5' to 3' or 3' to 5' is just the direction of the DNA. So that's how you refer. There's no north, south, east. It's just 5' to 3' or vice versa. So Okazaki fragments are the gaps on the lagging strand, and I'll explain what that is. The leading strand is where the DNA polymerase is creating new material on the old DNA. Lagging strand is the DNA polymerase having to catch up to the unwinding DNA section to create the new material, because it always goes 3' to 5' or vice versa. Okay, so now I'll explain the types of RNA. So as you can see, you can just read them on screen: transport, messenger, and ribosomal. So transcription is DNA polymerase that codes the RNA from the DNA. The RNA has coding regions called exons along the non-coding regions called introns. So what cuts off the introns are called spliceosomes, which cut off the unneeded regions. So it goes through splicing, capping, and polytailing of that new RNA. Okay, so translation. Small ribosomal subunits bind to the RNA that was just previously transcribed, and the small ribosomal subunit starts at the AUG start codon, a 3 base of nitrogenous bases that code for an amino acid. Methionine RNA binds to the AUG codon and a larger subunit to bind to the small subunit. The ribosomal subunit creates amino acid chain. This process is called elongation. When the stop codon is reached, the elongation project stops. Okay, so let's get to the type of mutations. So a point mutation is a change in one nitrogenous base. A deletion removes a nitrogenous base, and this causes a frame shift mutation. An insertion adds a nitrogenous base into the DNA sequence or RNA sequence, causing a frame shift mutation, and an inversion is a section of nitrogenous bases getting flipped with each other. Okay, so let's explore mutations a bit. So substitution, a silent mutation, is when the codon letters change, but the same amino acid is coded. Substitution, missense, is when the codon letters change, but it means a different amino acid. Substitution nonsense is when the codon letters change and it causes the code to end prematurely, as like a stop codon. A frame shift as insertion and deletion when the whole codon gets shifted over due to the letter change. Okay, now let's move on to Unit 7, Natural Selection. Okay, so convergent and divergent evolutions. Convergent is where a different species develops similar body structures over time to another animal. They're not becoming the same species. Now, divergent is when one species evolves into two different species, so they're becoming less genetically similar. Next is genetic drift, and that just means the changing allele frequencies by chance. So a common misconception is that natural selection and genetic drift are the same, but no. Genetic drift is just by chance. The animals just happen to be there and survive. Next, we got some math included into this unit, which is the Hardy-Weinberg equilibrium. So, it's two allele equations, two equations. The allele frequency equation and the genotype frequency equation. P plus Q equals 1, and P squared plus 2PQ plus Q squared equals 1. So first, by finding the allele frequency equation, you can find the genotypic equation, and then you'll be able to find the answer for the frequencies. Okay, now let's get to the evidence of evolution. So first, there's fossil records, vestigial structures. Like for us, we have wisdom teeth or the appendix, homologous structures, how similar structures are compared to each other, biogeography, their locations, and direct observations. Alright, so what you're seeing is called a cladogram, and this is to show the relations between species and their ancestors and where they split off. Think of the cladogram as a timeline, showing where species diverge. So lower is longer ago, and higher up is more recent, and that'll show where certain traits are. Now, a big part of this unit is speciation. It's the development of new species. Same species can interbreed and can produce offspring, but an allopatric barrier is a geographical factor stopping this interbreeding. Now, prezygotic and postzygotic so for prezygotic is a behavioral isolation where they just choose not to mate, and offspring are unable to reproduce in postzygotic. Okay, so we gotta know what causes variations in populations. Well, a variation can be caused by either genetic or environmental change. Variations have a lot to do with the natural selection and specific chromosomal differences. So if there's a mutation, that could cause something. If there's environmental change, that could make them adapt. An example of this is the physical boundaries causing environmental barriers. Alright everyone, last unit, Unit 8, Ecology. Now, let's learn about biotic and abiotic factors. A biotic factor is any living organism, like an animal or bacteria. Abiotic is when any non-living organisms, like rocks, water, dirt, that type of stuff, in an ecosystem. Now, the levels of organization. An individual, an organism, a population of them, a community of organisms, an ecosystem, a biome, and a biosphere. Yay! More math! Okay, so we gotta learn exponential growth versus logistic growth. Exponential growth is a population that grows exponentially, which is not really realistic. Now, logistic growth is population growing realistically, so it has a carrying capacity. And you're gonna memorize those two equations. Now, let's learn about the energy pyramid. Trophic levels show the passage of energy, so 90% of the energy is lost as heat when it's eaten by the next animal. So, an endotherm and an ectotherm. These are really simple. Ectotherms are cold-blooded animals, endotherms are warm-blooded. Okay, so let's learn what a niche is. It's a role an organism plays in an ecosystem. Competition is fight of organisms for limited resources. Keystone species are animals that an ecosystem needs or will collapse. Primary succession is an ecosystem forming from nothing, rocks and bacteria. Secondary succession is ecosystems forming after the destruction of a previous ecosystem. And we're done! Now remember, it's not magic. It's called science.